kind of left in a cliffhanger, didn't I? I was telling you about how you need to uh, take DNA and transcribe it to messenger RNA before you use a codon chart, and that you needed to know how to use the codon chart. And so we did TCG gets transcribed to AGC, and then we found that A, and then G, and then C, boom, that would be serine. And then I was trying to make the point about how if you've already got messenger RNA, don't flip it again, or you might get the answer wrong. And then I found it still would be serine. So the last thing that I left you with is that this system that was devised was not just devised kind of like a committee of people would have devised it. If a committee of people saw that there were 64 codons and there were 20 amino acids, that we probably would just have 3 codons for each amino acid. But that's not the way life has organized it. How has life organized it? Life has organized it so that really important amino acids like serine or like arginine, these guys get many more codons assigned to them. How is that helpful? Okay, well, let's look at arginine. Okay, no, let's look at serine. So with serine, if my DNA is supposed to be TCG, if it's supposed to be TCG, but I make a mistake and I make it TCT. Let's just imagine that happened, okay? So it was supposed to transcribe to AGC, but because there's been a mistake, it's, oh, I did it wrong. Let me try again. Instead of TCG, it's TCA, okay? So now when I transcribe it, instead of being AGC, it's AGU, right? Now, technically, when you have got a mistake in your DNA, that's a mistake, okay? And a mistake is a mutation. So there is a mutation there. But what was the impact of the mutation? Well, with the way the message was supposed to read, it was supposed to put the amino acid serine in the protein, but instead it put the amino acid serine. No difference. No difference. As a matter of fact, you and I, we have got areas of our DNA where you have got TCG and I've got TCA. But we make an identical protein. So which one of us has the mutation? Well, it'd be very difficult to figure that out, right? But it doesn't even matter because we're going to make the same protein, which leads me to a discussion of mutations. But let's practice this one more time before we go to a discussion of mutation. All right? ATA is going to be DNA. It transcribes to UAU. So the first letter is, go ahead and pause it, try and get it yourself. U, A, U, right? U, all of these start with a U. These guys right here, a U and an A. This one's UAU, that one's UAC, that one's UAA, and that one's UAG. Notice that there are 3 codons that have been set aside to say, stop, you're done making the protein, time to start over again. And there's only one that's set aside to be a start codon. All right. Let's talk about mutations. Right? I am going to slip in a new slide so we can talk about some mutations. Mutations. There are 2 kinds of mutations. There is a point mutation. Point mutation. And there is a frame shift mutation. Frame shift. That's a weird way to spell frame shift. Okay, frame shift mutation. Let's start with the sentence, the big red cat ate the rat. Great. The big red cat ate the rat. A point mutation would be if, oh, by the way, each one of these is supposed to be a codon. Codons are 3 letters, 3 nucleotides, right? So I'm going to change it so that I'm going to do a point mutation. And I'm going to change the letter B into an R. Okay. And now it says the big red cat ate the rat. The big red cat ate the rat, right? Now, does the sentence make sense anymore? Well, yeah, okay. You're going to be left wondering what a big red cat is, but you know it's a red cat, and you know it was bad for the rat. So you basically have the meaning of the sentence. This is called a point mutation. Why is it a point mutation? Because at one point in the DNA, there has been a substitution. Point mutations, there has been a substitution of one nucleotide for another, right? Now, remember, let's go back here, back, oh, it got erased. Back here, if I, instead of having TCG, if I had TCA, that would have been a point mutation. And TCG is AGC, and AGC is serine, and TCA is AGU, and it's still serine, right? That would be an example of a point mutation. Now, point mutations are not always benign, not always. Okay? This particular one that we're looking at, it did change the meaning of the word, right? It's a rig instead of big, you know. Now, whether that will destroy the protein, like I can't make brown pigment in my eyes, or whether it'll just slow the protein down, or maybe not change the protein at all, that'll depend on that whole transcription thing. Okay, but point mutations. Sickle cell anemia is a very important medical problem, and it is an example of a point mutation. However, it's kind of the exception to the rule. Point mutations in general can be very survivable. Let's look at a frame shift mutation. In a frame shift mutation, instead of just substituting one nucleotide for another, you end up either adding or removing a letter. So instead of substituting a letter, I'm adding the letter. Now what happens? Do you see what's going on? I still have codons that are 3 letters long. When I added one, same thing would have happened if I subtracted one, everything gets jostled, right? Okay. This is a frame shift mutation. A frame shift mutation is the insertion or deletion of a nucleotide. And if I ask you which one is more survivable, point mutation. Because you can get a point mutation and literally have no difference in the protein at all. Frame shift mutation, that's almost never going to happen. They're bad. Frame mutations are bad. Right? Now, this will be a short video. We're going to do a short video on the last part of our lab. Okay, one more.